Welcome to this very special lecture over a very long distance. I'm glad that I'm able to share a little bit the results and thoughts that we have here in Lausanne with you. I'll now switch to the shared screen, and this will just take a minute. And let me go full screen and select the laser pointer. And here we go. So the topic of my talk is eligibility traces and three-factor learning rules. And it's joint work with many people, and it's not only work from my own lab, but it's built on a long tradition around three-factor rules about plasticity, about learning. Well, yes, I'm interested in learning. I'm interested in memory formation. Think of, if you take a train, there's a stream of inputs if you look out of the windows. Do you really want to remember all of the trees that pass by, all of the houses that pass by? No, not really. There are very special moments, often emotional moments or stress moments or traumatic moments, which you will in the end remember. So this is the first aspect. How do we remember? The second aspect, well, memory, we also have memories of skills, memories of skiing, of biking, of playing table tennis. How have we learned these skills? So how do we learn? Now, we believe as neuroscientists that the learning that we see in form of behavior is linked to neurons in the brain, which are connected to each other by synaptic contact points, by synapses. And these synapses have a certain strength, which I can measure by sending in a spike to the synapse and measuring the response. And now the important point is that this response strength, the strength of the synapse, the strength of the connection can change. And that phenomenon is called synaptic plasticity. And so the idea is that learning, whatever we learn, is linked to synaptic plasticity. So I would like to go over some very classic work related to Hepkin learning and spike timing dependent plasticity. And then I will go to the specific generalization of three-factor learning rules. Again, it's a framework, it's not a single rule, before we come to a couple of examples. Let's start with Hepkin learning and SDDP. Most of you will have seen this kind of statement of Hepkin's learning rule. I have two neurons that are connected to each other, and what matters for the connection strength from neuron J to neuron I is the joint activity. The presynaptic neuron, the sending neuron must be active, the postsynaptic neuron must be active. If the presynaptic neuron takes part in firing the postsynaptic one, then some change happens. And here, the quick summary usually is stated as, if neurons are active together, their synapse is strengthened. The synapse between those two neurons that are linked is strengthened. And of course, there's a long tradition of experiments. There are excellent reviews out here. Let me explain a little bit how we think about Hepkin learning. So I have two neurons now that are active together, and these two neurons, the presynaptic will send out a spike, the postsynaptic will maybe send out a burst of spikes. Then if the two are active together, then this specific synapse, the green one here, is strengthened, but not the red one. But joint activity doesn't need to have spikes on the side of the postsynaptic neuron. It could also just be a slight depolarization of the postsynaptic neuron. As a mathematician, as a theoretical neuroscientist, I want to formulate these changes, the change as a function of time, well, as a function of what's happening. And now the Hepkin learning rule tells us What matters is the firing rate of the presynaptic neuron, the sending neuron, neuron J, as well as the firing rate of the postsynaptic neuron. And then this synapse here also knows its own weight. So these are the three possible entries if you think of a rate model of synaptic plasticity. A synapse can only use locally available information, and that means it can only use its own weight. It can use the rates of the presynaptic neuron and of the postsynaptic neuron, but it cannot directly use the rate of some other neuron K. I'm a theoretician, and I don't know this function f. So what I do is I make a Taylor expansion, and then we have a first term that just says, well, spontaneous changes can happen. If the presynaptic neuron is active, then additional changes may happen. If the postsynaptic neuron is active, additional changes may happen. If the two neurons are active together, additional changes may happen. And often people say that this term is the Hepkin learning term. Now note that these expansion coefficients can still depend on the weight, the momentary value of the synaptic strength. So here's the formula again. We have these different terms. Now I would like to call all terms that have a pre and a post together as, I would like to call them Hepkin terms. And this is not the only one. There's another one here, and there might be further ones coming later in the expansion. Then we have other terms that only concern the postsynaptic firing rate, and this leads to heterosynaptic plasticity, because not just this synapse from J to I is changed, but also another one from K to I if this postsynaptic neuron is, for example, highly active. And then we have other terms that only depend on the presynaptic activity, and I call them transmitter-induced. And of course, this series doesn't end here. This series may continue and contain many more terms. So here is the classic Hepkin term. This is another Hepkin term. Here is a pure post term, and there are other pure post terms, and then we have the pure pre terms. So now the question is, what is a Hepkin learning rule? And currently we just stick to rate models. So people say this is standard Hepkin learning. If you look at the Wikipedia article, this is what you see as the first example. Now let's think of a neuron that's highly active, a neuron that's on, say, it has 20 Hertz activity. The postsynaptic neuron is on, then 20 times 20. This gives a positive change, but if one of the two neurons is 0, it gives no change. And now you see immediately that this rule is a bit strange, because you can only increase, but you can never decrease a synaptic weight. But there are simple ways to allow for this. The first one is we just use this spontaneous activity term with a negative sign. So now it's minus C, and with this minus C, I have solved the problem. Fine, but this is not the only possibility. But we could also use a formulation like this, say, highly active, an active neuron means 20 Hertz. I compare with a threshold, say 10 Hertz, and then you get this kind of configuration. If the two neurons are on, it's fine, it's positive. But if the presynaptic neuron is off, nothing happens. If the postsynaptic neuron is off, but the presynaptic is on, I get a negative change. And then there are other combinations, like this one here, where again you would have just a Hepkin term, but now it's quadratic with the postsynaptic rate, not just linear. Now all four of these are Hepkin models. All four are local models because they only use the information that's locally available at the synapse. But these are not the only models. There are many other models possible. And therefore, we can say it's not that there is a Hepkin learning rule. It's not the one Hepkin learning rule, but it's really a family of different learning rules. So Hepkin learning is not a single rule. Hepkin for me is sort of all local rules where at least one term has this form here, a combination of presynaptic activity and postsynaptic activity to some arbitrary power. Could be linear, could be to the power of 3 or 4, and so forth. BCM learning rules fall in this class. But then Hepkin learning may also contain other terms, the pure post terms. It may contain pure pre terms. And when I say pre and post, I don't mean it's implemented on the pre or postsynaptic side. I mean, it depends. The change depends only on the pre activity or only on the post activity. Now, suitable combinations of these terms enable formation of memories, of assemblies in networks. It enables modeling of spine dynamics. And in my lab and in many other labs, many groups have looked at suitable combinations of terms that would allow this. So this is for rate models. Now, firing rate is not all, because real neurons fire spikes. So now, what's the generalization of this for spiking models? Well, I have this presynaptic spike train here. I have a postsynaptic spike train. I have the postsynaptic potential, because it's not just that pre and post spikes together do something, but it's also presynaptic spikes together with postsynaptic depolarization that do something. Okay. Now, the simplest model of spike timing dependent plasticity is the following. The presynaptic spike leaves a trace, say an exponentially decaying trace. Then the postsynaptic spike comes in, and if the postsynaptic spike comes in after some delay after the presynaptic one, then it basically just reads out the momentary value at the moment of the postsynaptic spike, and this gives the weight change. And the amount of weight change may depend on the momentary value. So if the weight is already big, it might be difficult to make it even bigger. And so this might contain some saturation. If the weight is small, the effect might be larger. This is just the differential equation describing this exponentially decaying trace. At the moment of the presynaptic spike, I get the jump upwards, and afterwards it's just a decay with some time constant. But now the postsynaptic spike also leaves a trace, again exponentially decreasing, and the later presynaptic spike will read out the value of this other trace. And if you put this together, then you get a standard STDP model where you get LTP for pre before post timing and depression for post before pre timing. So this is now our Hepkin term. The presynaptic spike leaves a trace, and then you need the postsynaptic spike to implement the change. So STDP is a spike-based version of Hepkin learning. Again, it's not a single rule, but a framework. What I described were just a pair of spike, one presynaptic, one postsynaptic, but you can now do the same kind of expansion, and you would find that a presynaptic spike compared with a pair of postsynaptic spike explains experimental data even better. And we could also terms that are homosynaptic, that are heterosynaptic, that are presynaptic only, transmitter induced only. So the whole family of learning rules can also be formulated with spikes. So again, it's not a single spike timing dependent plasticity rule, but it's a family of rules. This is now a very general form of Hepkin learning. Pre could stand for spike arrival at the synapse. Post could stand for back propagating action potential. for voltage at the location of the synapse, or some trace left by the voltage, such as calcium elevation, some second messenger. The pre might be the presynaptic spike, but it could also be just the neurotransmitter in the cleft, or the amount of neurotransmitter bound to the postsynaptic potential. So Hepkin learning is not a single rule, it's a very large family of rules. And so one example, you can use this to describe structural plasticity and also account for a very peculiar effect. If a neuron J is connected to a postsynaptic neuron, then it usually does not make a single contact point, but it makes 4 or 5 or 6 contact points. So if you look at the fraction of connections with the number of contacts, then most pair of neurons, take neurons with a distance of 1 millimeter, most pairs are not connected, but if they are connected by 5 or 6 or 7 or 8 contacts, but never by 2, very rarely, or never by 1. And now you can build a model in that framework, a spiking model, a STDP model for structural plasticity that accounts for this distribution. So if there's a connection, then there are 4 to 6 contacts. And then one can follow this over time. Suppose we have a model where we have a few hundred spiking neurons for barrel cortex, first column, barrel cortex, second column, barrel cortex, third column. We have different inputs from thalamus. And now one of these inputs is lesioned. Then these Hepkin learning rules, this family of Hepkin learning rules accounts well for the fact that the input restructures, so that 3 days post-lesion, the input that went previously to column 3 is now integrated into columns 2 and 1. And so neurons that have previously been assigned for taking care of this input are now partly integrated in column 2 and partly integrated or take over tasks of column 1. You can also, with this kind of model, account for the spine statistics. So the lifetime of spines has been measured in different papers. These are just acronyms, papers of Holtmar and Löwenstein and many others, and Young. And the model very, very nicely accounts for this behavior. So the model is the solid line down here. So, first question, how do we remember? Well, how is memory generated in synapses? We believe that Hepkin learning is a good candidate, and Hepkin learning is not a single rule, it's a family of rules. It's a very, very large family of learning rules. And this can be used to account for formation of assemblies after presentation of specific patterns, or it can account for spine dynamics and structural plasticity. Okay, so now we have this very, very broad, large family of learning rules. And now the question is, well, if this is so general, we should be able to do everything in that framework. So the next question is that correct? Is that true? And for this, I will now turn to 3-factor learning rules, and this will be the main topic for today. Let me just take a bit of water. So let's start. These are the 3-factor learning rules that we'll consider now. We said Hepkin learning is a good candidate for lots and lots of things, but how about this? How about learning skills? How about learning table tennis, skiing, biking? How do we learn? Can we write down good learning rules for this kind of tasks as well? Now here is the outline of one of these tasks. I have a little mouse sitting in a corridor. At this location here, it can turn right, or it can turn left. And if it turned left, it would get some Swiss cheese. Now we know that there are place cells in hippocampus. So if the mouse is at this location, some subset of neurons would get active. The mouse in the first trial decides to turn to the right. So some neurons in some brain area, I don't specify which ones, would get active. And then the motor plan would be carried out. Suppose now we have Hepkin learning. Now you see I have neurons that are connected with each other. These neurons are jointly active, so these connections would get stronger. But is this what we want? Well, not really, because the mouse had turned to the right. If I now strengthen these connections, the next time the mouse is at this location, it would be even more likely to turn right. But this was the wrong action. So, how can we generalize Hepkin learning to account for this? And here's the idea. You don't change the connection when you see joint activity, but if you have coincidences of pre and post, you just keep an eligibility trace. And this eligibility trace would normally decay over a few seconds, unless something else happens, unless we get a success signal. And the candidate for the success signal would be dopamine. Now, suppose in the first trial, the rat took the way to the right, in which case it would not get the success signal. Suppose in the next trial, it turns to the left, in which case it gets the success signal. And now these connections are strengthened. Whereas the first one just kept an eligibility trace which decayed and was not strengthened. So the strengthening is due to this extra signal that conveys the notion of success or of reward. Success for me is reward minus expected reward. And if you think about this, if you look at the book of Sutton and Bartow on reinforcement learning, it contains algorithms of TD learning, temporal difference, of SARSA, of policy gradient. All these algorithms can be formulated in the framework of three-factor rules that need eligibility traces and a success signal. And now I will tell you a little bit more how these algorithms work. So what's this success signal? For me, the success signal is a more or less global signal. Dopamine is one of the examples. It has alborizations that go through large areas of the brain. Noradrenaline has a very similar broadcasting behavior. Now, often it's said that dopamine signals success in the sense of reward minus expected reward. However, it's not necessary that dopamine and success are perfectly aligned. Suppose that I have this rotation, I have this slight shift of the axis between dopamine and success, and say between surprise and noradrenaline. Then the two together, noradrenaline and dopamine, together would still span the space to account for reward and for surprise. So here's the basic idea, how these three-factor rules work. I have my two neurons, they are connected to each other. The first one sends out a spike, the second one sends out a spike. Now, I just set a flag, I leave an eligibility trace, and then we wait for the success signal, and then the synapse is strengthened. So you see that I first talked about Hepkin learning rules. And now I'm saying, well, this in a way is not enough because I need the success signal. I need the third factor. Now, what could this third factor be? A standard example is dopamine, but acetylcholine, noradrenaline, serotonin are other candidates that have this broad spreading, the broadcasting of some global signal, be it success, be it attention, be it stress, be it novelty or surprise. Now let's work on the math just a little bit more. One specific instantiation of this pair would be pre, before, post. Great. This gives STDP, but only now with a success signal. So now we say the STDP increases a eligibility trace, but this normally decays with the time constant tau, unless while the flag is still there, during a few seconds while the flag is still there, we get the success signal. And then this really changes the weight. And this has a long tradition in computational neurosciences, and I'll just give a couple of references here. So the strengthening of this connection needs three factors: the presynaptic activity, the postsynaptic activity, and the success signal, the global signal, the third factor. So I started off with this kind of graph. Now we add an extra panel where we say pre and post together leaves a trace, but it's not enough for a change. We need a delayed neuromodulator signal. Now, why the delay? Well, think of the little mouse that has to turn left. It will find the cheese only with a delay. Think of the table tennis player that trains up to learn a new serve. Well, whether the serve is successful, he will see only a few seconds later. If you think of a baby that learns to grab an object, it will do a couple of movements next to the object before it's finally successful. You don't want that all the wrong movements get reinforced. You want the one movement that leads to the success to be reinforced. Now, if you think about this, the essential grasping movements are in the range of 1 second, maybe 2 or 3. The table tennis feedback is in the range of a few seconds. The mouse feedback is in the range of just a few seconds. So you would expect that a delay of a few seconds is about right to give rise to a reinforcement of these synapses. So if you want the prediction, but it was never formulated as a prediction. So the way modelers set up their timescales was that in order to explain the behavior, they had to say, well, the neuromodulator should work if it comes with a delay of 1 to 5 seconds. And much, much later, this was actually measured. Here is the first experiment that I know that has this nice temporal setup. So this is pre and post together, pre, post, 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 pre, post, post, post a couple of times. The pre-post signal ends here. And now the dopamine can come with a delay of 1 second, and it causes a change of the synapse. Of course, it comes a bit earlier, it's even better. But a second or so doesn't hurt. Then the following year, there was another experiment, and this is now with serotonin and norepinephrine, and the signal is kind of the same: pre, post, 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 and then a delay of 1 or 2 seconds still gives with 5-second delays and norepinephrine, you still see a strong increase of the weights. With post before pre and a signal of 2.5 seconds, you see a strong depression. Message here: yes, the neuromodulator can come with a delay of 1 to 5 seconds. The third experiment is a bit different. Here it's in hippocampus, and there's a burst of presynaptic spikes which causes just the postsynaptic depolarization. And then with a delay of 1 or 2 seconds comes a burst of spikes, a dendritic potential. And if these pre and post here, and now this takes the role of the third factor, if they are together, then you see the increase in the weight. So this dendritic spike is also called a complex spike, and this can come with a delay of 1 to 5 seconds. Now, often I get asked, well, these global signals, isn't that way too unspecific? Well, let's just walk you through a simple example. It's completely made up. It's not based on any specific experiments, just a thought experiment. So I have here a pair of neurons, and neurons come in two types. They are the orange neurons and they are the blue neurons. So now this is orange to orange. The two get active together, so it sets a flag. And then I get one neuromodulator and the synapse is strengthened. Now, I could have the two orange neurons active together, it sets a flag, but now I have two modulators together, modulator 1, modulator 2, and something else happens. For example, LTD. Or I could have an orange and blue neuron. They get active together, and this gives LTP with the two neuromodulators. Or I can have blue to orange, and it gives no change, and I can make many other combinations. So the point is just the combinatorics is such that with just two neuron types and two neuromodulator types, I have many, many possibilities. Now think of 30 neuron types and 5 neuromodulators, and you can believe me that I can get a very specific signal to a subset of the synapses. So, summary of this part. The 3-factor rules are a framework. It's not one rule, it's a framework. It has always 2 local factors, pre and post, 1 global factor, sometimes several global factors. It's global in the mathematical framework. In a sense, it's the same for many neurons. Doesn't have to be completely global. Dopamine doesn't get to visual cortex, for example, but that doesn't matter for the sake of the following arguments. Now, the global factor could be reward minus expected reward. It could also be surprise. We can generalize to several neuromodulators and get out of reward to surprise, to something exciting, good, whatever emotional brain state could play the trick. So now I would like to walk you through 2 examples very quickly. The first one is learning mazes. So it's like the T-maze we had for the mouse. It's now a maze with an obstacle here. There's a goal here. We have potential starting positions. So it's like a Morris water maze with 4 starting positions, but with no obstacles. And the agent does not see the location of the goal. And now we use these learning rules that just have pre and post and success, and we do this with spike and neurons. So what are the assumptions? Well, the assumptions are that we have place cells. Each place cell is sensitive only in a small region of the environment. Then we have a ring of neurons that play the role of the actors. And both of these represent a continuous state space. I have spiking cells, but the location here and the location here and there, everything is continuous. It's just coarse coding, different subsets of neurons go on. Same thing here. For the actions, I have a continuum of different actions. I can go north, northeast, north, northeast, north, north, northeast at an arbitrary position. And then we have a third group of neurons, and these are neurons that code for the value, the momentary value of the position. And that value, of course, itself is also learned. So overall, this is called an actor-critic architecture. This is the action part, and this is the critic part. And now we use the same 3-factor rule for these connections as well as these connections. And the idea is that these neurons calculate the value, and this allows to get a temporal difference error, and this will influence the 3-factor rule here and there. And so this is now the result. So in the first trial, this is dark blue, the red, the mouse, the agent just wanders around and it finds the target by chance. But after something like 20 or 30 trials, you see these three-factorial rules in an actor-critic architecture work very, very well. And this is, after many more trials, this is the value map that develops. So being here, you already know that you will approach the goal this way. Now, if you look in the literature, and if you have something in mind, you can always find some papers that show something similar. So with our value neurons, we would predict that the closer you get to the goal, the higher the value. And indeed, there are neurons in some brain area that have this kind of behavior. So the value of a position is a very important concept in temporal difference learning, TD learning. And the value in our network is represented by neurons that are part of the critic of this extra population of neurons. So let's see how this could work. And this is inspired by recent experiments of Watabe Uchida. So the idea now is that we have these value neurons that we have explicitly modeled, spiking neurons. But these neurons should actually communicate with other groups of neurons that have the value of the previous time step so that the two together can calculate this temporal difference, indicated here by a dot. And then you have to compare with the reward, and then this gives the delta signal. Delta is reward plus the value, the position now, minus the value of the previous position. This delta error or TD error is an important concept in a large class of learning rules. So the three-factor rules are a framework. I didn't say that much, but it didn't matter whether we used an SDDP rule together with the global factor, or whether we used a variant of SDDP where we only have pre before post, but no post before preterm. The details don't matter. What matters is that we stay in this framework of three-factor rules. And in that framework, it's very important that the global factor is the TD error and not just the reward itself. Otherwise, it wouldn't learn. The reason is that I said that the timescale of the eligibility trace is in the range of 1 or 2 of maximum 5 seconds, but the movement of the agent towards the goal may take 15 seconds or 20 seconds. So you need this TD error so that you can propagate information about the presence of reward to locations that have been visited more than 5 seconds before. But the timescale of the eligibility trace used in these simulations was exactly the one found in the experiment. In fact, the simulations were done before the experiments were done. So I already spoke about timescales. I spoke about eligibility traces. I showed examples of experiments that were done in different brain areas. Now the question is, and this might be interesting for some of you, do humans show signatures of eligibility traces? Now, of course, I cannot measure, I don't want to measure inside the human brain, but there are markers. And let's look at what global markers could indicate. So here's the basic behavioral experiment. We have a start position, then we do several actions before we arrive at the goal. So we want to set up an experiment, a psychophysics experiment with that layout. Now, suppose we initialize all the values in a model of all these states at 0 or just at the same value. At the beginning, before the first trial, you don't know what you see, so there's no value attached. You don't know where you are. So as we move along, well, the value is 0 here, the value is 0 there, the reward is 0. 0 plus 0 minus 0 gives 0. Okay? There's no delta error nowhere. Now this means that in the first episode, nothing is learned except for the final step, because this is where the reward happens. So 1 step before signals arrive, but 2 steps before, delay 2 before, no information about the goal can arrive after the first episode. Now, this is the trick that tells you how to design an experiment to test eligibility traces. Without eligibility traces, learning is slow. 2 steps before the goal, you haven't learned anything. With eligibility traces, of course, it's different, because then what matters is, well, whatever you have seen during the last 5 seconds is in your eligibility window. So standard TD learning without eligibility traces is slow. Now let's check how humans perform. So we performed an experiment using clip art images that represent 10 different states. You can play the experiment yourself in the following, supposing that the video works. If I can make it run, you will see the first image now, and say you want to take the left action. That means you put your hand down on your left knee. Take the right action, you put your hand down on your right knee. That's your decision. Okay, and here we start. Okay, this is the goal. So I hope you have taken actions after each image. Second episode right now. Have you seen this image before? Well, would you take the same action? Now, make your decision. So the design of the experiment is as follows. In the first episode, you saw this sequence of images. In the second episode, we arrived here. And now the question is, will you take the same action again? Now, this was an image, the cup was an image 2 steps before the goal. So without eligibility traces, the prediction is you have not learned anything. If you have the inclination to say, well, I saw that image before, I would take the same action again because it was actually good, then you show a signature of eligibility traces. And so here is our hypothesis. In state D1, 1 step before the goal, of course, you always learn, but in state D2, without eligibility traces, you wouldn't learn. But with eligibility traces, well, you took action A, you would take action A again. Okay, so, and this is how humans perform. Actions in state D1 have a strong bias that indicates, yes, the experiment works. People know what they have to do. But importantly, in the second episode, they show a strong bias to repeat the same action, also in the state delay 2 steps away from the goal. And that means they show a signature of eligibility traces. We call this the behavioral signature of eligibility traces. So this means we have one-shot learning. It's enough that you take an action a single time. It's enough that you see the target once, and that bias you in the second episode. So one-shot learning is compatible with eligibility traces. And then if we fit a model, then we see the eligibility traces should be in a range of 10 seconds, which is the right order of magnitude It's not exactly the same value, but it's a different, it's humans, it's not rats, it's a different brain area, so why not? So this is just behavior. Now, I said we cannot look really inside the brain with our human subjects, but we may still want to see some physiological signature. And here is the prediction that if you find a signature, then if there is no eligibility trace, you should have no effect in stage 2. With eligibility trace, you should see an effect. Now, the signature we take is pupil diameter. Now, of course, we know that it responds to light intensity. Many of you know that it also responds to memory load, to engagement, to surprise. More generally, it's known to be a learning-related signal. And so we measured this, and what we see is that in episode 2, the red one, the response is very different from the black one, which is episode 1. And that's true in both state D1 and in state D2. So the pupil diameter shows some signature of temporal difference learning with eligibility trace. We made additional checks that if you just show the same sequence of actions, or if you let them do the same sequence of actions in other conditions, then you wouldn't see this change of eligibility, this change of pupil diameter. So the decay time of the eligibility traces that we see is in a range of 10 seconds. It's right order of magnitude. The reinforcement learning models make qualitatively different predictions, whether they have whether they are models with or without eligibility traces, and the experimental data is in favor of the eligibility traces. And the reward a few seconds later, therefore, influences state actions associations, which is consistent with the 3-factor rule framework. It's not a proof that it's implemented this way in the brain. I'm just saying it's consistent. The behavioral and pupil data is consistent with this family of 3-factor learning rules. So let's summarize and maybe have a little bit of an outlook. I said that the learning framework with 3 factors is general. It's not linked to success in the sense of reward minus expected reward. It could also be surprise. Now, I hope you will enjoy these images, and let me just try to run this. And I hope that I can start the video. No, the video doesn't work. I will show it separately a bit later. So the point of the video is that even if you don't know the images, if you just look at new images, at some point you are surprised. And the surprise is not linked to the novelty of the images. It's something else. And in the absence of any reward. And the idea is now that instead of modulating learning by reward, we can modulate in models learning by surprise. And for this, we have to work on good definitions of surprise. And surprise is not novelty. Surprise is something against the expectations. So you have to expect something to be surprised when you see something else. And then this links to predictive coding. We have to ask the question how the brain calculates surprise. And we are currently working on all 3 of these questions. Let me conclude. In the review part at the beginning, I wanted to show that Hepkin learning is a family of learning rules, but synaptic plasticity is more than Hep because it also needs the 3rd factor. And this 3rd factor could be implemented by neuromodulators that account for reward or surprise or other emotional states. The time scale can be measured. It's 1 to 5 seconds. It's a few seconds in the slice experiments. In our psychophysics experiments, it's a range of 10 seconds. And it's one-shot learning. Reinforcement learning can be very, very fast. Learning can be done in a few trials. Sometimes you hear that, wow, reinforcement learning is so slow, it's so slow. No, if it's set up in the right way, if you have place cells that represent the environment, then actually it can be very fast. Now, just a very short historical remark that links to the interactions between theory and experiments. So three-factor rules are a conceptual model. And the first paper I found is from Crow in 1968 and just describes it in words. Well, you need this kind of confirmation signal. Learn now. And then Klopf and Bartow and Sutton really worked this out in the first neuronal model. Then they went into TD learning. Other people worked on TD learning. Then it went a bit back to the biology for Moritz Watermaze experiments, reinforcement learning. Then people started to put in STDP, they put in spikes. And finally, we have a model where everything is put together. And around 2015, the first experiments were done. Now, many of these models used three-factor rules. Many of these models use timescales of 1 or 5 seconds. Nobody said, well, this is the way the brain has to implement it. It's not a strict prediction that it has to be that way, but it was just very, very convenient for theoreticians, for modelers to describe it this way. And so, yes, it's a conceptual model. It's not, it doesn't predict the value doesn't predict specific numbers. But this kind of conceptual models are those that actually, over the long term, over 10, 20, 30 years, do influence the thinking in the experimental sciences. With this, let me end. The work is based on collaborations with many people, with many former PhD students that now are at different places. And for some of the results, you also find a summary in the textbook on neuronal dynamics. With this, I would like to end. Thank you very much for your attention. And for the question part, you can address your questions directly to me. Thank you very much.
That's valid. I think for a large family of these three-factor models, that the reaction time will always move as early as possible. So the models will give this automatically. If you have this kind of STDP models, then they would have this forward shifting in time, which would explain the fast reaction time. But only on a very conceptual level, I cannot give any specific numbers. But in general, the phenomenon is well known.
Thank you so much. Another question is written in the question part. I read it for you. In some diseases, dopamine release is affected. Is it still a good measure for success?
Yeah, that's a long-standing debate. So I had this one graph where I had this rotated system between success and dopamine and acetylcholine and maybe serotonin. So there's no need to have a perfect one-to-one alignment with dopamine and success. That's the first part of my answer. The second part is there's a difference between phasic dopamine signals and tonic background signals. And the signal I use as a success signal would correspond to the phasic dopamine signal, or in view of the shifting of the coordinate system, it corresponds to a phasic signal in some combination of dopamine and acetylcholine and serotonin or whatever. So that's my answer.
Thank you. Thank you so much. Is there another question? So
seems not.
I have another question. In your presentation, you emphasize in the role of the third factor in the learning, and actually you used some Q-learning models like temporal difference model for modeling of these. But I have some question about the fitting procedure. Actually, in the fitting procedure, we don't use a biological procedure for fitting the model and the data. So how can we explain that this model is really present our behavior?
Are you referring to the psychophysics model where we had the decision after in the image sequence?
Yeah. Yes.
Yes. For that, the weak. So we have no proof whatsoever that what we see in the behavioral experiment must be explained by a three-factor learning rule. So you're absolutely right. So the fitting of the behavioral data is in the context of some TD learning, Q learning with eligibility traces, and then we extract a time scale, but we don't say that it's implemented as an eligibility trace. Of course, I put this in the same talk of the eligibility traces, of the synaptic eligibility traces, because I believe that this could be the case. But there's no need that it should be a synaptic eligibility trace. The memory could be some other form of memory, a non-synaptic transient activation memory in neurons, rather than sort of this hidden synaptic flag variable. Yeah. So yes, we have behavioral data and we fit a model, and other people have fitted different similar models to similar kind of data, but that on its own is no guarantee that this model is implemented by a learning rule on the synaptic level. I do believe, or I want to believe, that this is the case, but there is no scientific reason that it must be this.
Okay, thank you so much for your answer. There is another question in the question part. I just read it. Does surprise have the same definition as Shannon information?
The answer is no. You want more? So I don't know how much time we have.
Approximately 20 minutes.
Okay, so let me try to share the screen and I'll show you the surprise video that didn't come out. Do you see the screen now with the images?
Yes, they're here.
Okay, and now it should work. Okay, sorry. Maybe I have to go. Stop. Let me try to do it full screen. Now start the images. No, it works. Hopefully, no?
Yes,
Okay.
we are seeing it here.
Okay, just watch the sequence. Okay, that was it. In total, it was, so it didn't run completely regularly, yeah, and it's not in the form of a controlled experiment. But you saw 15 images, and some of them were the same, okay? But some images you saw for the first time. Well, you have seen none of the images before because we created them. There are also these fractal images or fractal art images, sometimes called. And the first image was new, the second image was new, the third image was new, and the fourth, I think, and then we went into the intro loop. And then at some stage there came one additional image that was also new. And if I ask online in the audience whether they were surprised, they were not surprised by the first image, they were not surprised by the second image, but they were surprised by this one extra image. And why? Because we always build model about the world. So the surprise is not about the how unlikely, how improbable an image is, because image number, when you saw the image number 2 for the first time, it was completely new to you. And if you saw this extra image for the first time, it was completely new to you. But at the beginning, you didn't have expectations. At the end, or in the middle of the video, you started to build up expectations. So surprise is against expectations. And Shannon surprise does not include this. Shannon surprise does not take into account the expectations. Now there's also another definition of surprise, which is called the Bayesian surprise. And the Bayesian surprise is basically, it takes into account your own model, your own sort of view of the world. But Bayesian, and then Bayesian surprise is defined as how much you have to change the parameters of the model. So include the new perception in the new image into your model. And that's in a way a nice definition, but it cannot be used as a definition for learning, because basically means you first have to understand and include it, and then you know that this was surprising, but then it's too late. So it's too late to use it as a learning signal. So Shannon surprise would work as a learning signal, but it's measuring the wrong thing. Bayesian surprise, it's maybe measuring the right thing, but it's not useful as a learning signal. So what we are doing in the lab is we try to get mathematical definitions that allow to calculate surprise rapidly so that we can then use it as a third factor, and yet have surprise in this concept that it's against expectations. And that's the challenge, and we are working on this. So that's my answer.
Thank you so much for your complete answer. And is there any question? So, Mr. Farhang?
So no, there are no other questions.
We can finish. I think that finish the session.
Okay, thanks very much for the invitation. It was a pleasure to do it in this online format.
Thank you for joining us today.
Yeah,
It was
sure.
an honor having you here.
Thank you.
And
Thank
please,
you very much.
please enjoy the continuation of the meeting. Bye-bye.
Goodbye.
Thank
Thank you and goodbye.